0 as no change from the reference, and a 1 as a change from the reference. But in general, these labels could be arbitrary. We can represent the data as a k by n binary matrix. And in this small example here, we see that k would be 4 and n would be 6. A maximal perfect haplotype block is a subset of these sequences, the rows, and an interval of SNPs, the columns, that is completely conserved amongst the subset and cannot be extended left, right, or by adding any other sequences. Kanha et al. Formalized this definition using 4 conditions. The first says that all the sequences in the block must be equal, and the next 3 ensure that it's maximal. They also define notation that we'll use to indicate a block as a list of the sequence indices, then the starting column, and then the ending column of the block. So let's take a look at some examples to make sure that this is clear. First, do sequences 1 and 4 from column 1 to column 5 form a block? We can see that they satisfy the equality condition, and they can't be extended left since they start at the first column, so it's kind of trivial in this case. They can't be extended right because column 6 has a 1 for sequence 1 in column 6, and column 6 has a 0 for sequence 4, so they couldn't retain equality. And we can't add any rows without breaking equality. So it is a block. It is a maximal perfect haplotype block. Now, what about sequences 1 and 2 from starting and ending at column 4? In general, there's no problem including a single column as a haplotype block, and we can see that this one satisfies the equality condition, and we can look left and look right and see that it satisfies left and right maximality, but we could add sequence 4, and so it's not a block. Now that we've defined what a maximal perfect haplotype block is, what we really care about is how quickly we can find all of them from an input data set. When Kanha et al. First defined the problem, they gave a big O of kn squared algorithm to solve it. And by 2019, Belenko et al. Gave an optimal solution, finding all blocks in the size of the input matrix, big O of kn. They actually gave 2 solutions, the first using a suffix tree, and the second using a Burrows-Wheeler transform, positional Burrows-Wheeler transform. There they process the data column by column, and they only need O of k time for each of the n columns. And we'll talk a little more about this after we define the wildcard haplotype block problem in order to see how this method kind of breaks down with wildcards. We can now define maximal perfect wildcard haplotype blocks. We use a star to indicate a wildcard SNP, which we think of as being able to match to either a 0 or a 1. Again, we have case sequences of length n as input, but you'll notice that we've changed the equality condition to the consensus condition. We assume that each column of a block must contain a non-wildcard value. For example, in the data set here, the block shown contains at least 1 0 in the first column. It actually contains 3 zeros in the first column, and at least 1 1 in the second column. We say that 0 1 is the consensus pattern for this block. Oh, should I be? I realize I should be hiding this, huh? Sorry. The consensus pattern for this block, because all sequences of the block can be made to agree with 0, 1 by setting wildcards appropriately. For example, if we take a look at sequence 1, there's a 0 in the first column and a wildcard in the second column. So the sequence matches to 0, 1, the consensus pattern, as desired. On the other hand, A block containing sequences 1 and 2 at columns 2 and 3 does not satisfy the consensus condition because it does not contain a non-wildcard value in all of its columns. In fact, it doesn't contain a non-wildcard value in any of its 2 columns. But if we were to add sequence 3, then it would be a block because now it satisfies the consensus condition. And it can't be extended left, right, or by adding any rows. Without wildcards, there could be only big O of kn blocks. What about when we add wildcards? We use the symbol T to mean the number of blocks in a given input. We can give one bound on T by counting up, for each column 1 through n, the possible number of blocks that could end at this column. The proof comes out of the data structure that we'll discuss in our solution to finding the blocks. We do note that this bound does not appear tight. Our algorithm, which uses a trie data structure, is output sensitive in the number of blocks that do exist in the input, taking big O of kn times t time. A trie, so named because it can be used to efficiently retrieve the strings it is built over, is a tree data structure where edges or nodes are labeled with letters and paths from the root to the leaf. The leaves are words from the input dataset. In our case, the words are these snip call sequences, or the rows. Our algorithm, just like the algorithms proposed by Kanhai et al. And by Alenko et al., uses the following key observation. At each column, left maximal blocks correspond to internal branching nodes in the trie built over the reverses of the sequences up to that column. And so, to find all blocks in the input, we can iterate through each column, build a trie of the reverses of the sequences, and find the internal nodes. Alenko et al. Achieved optimal runtime using this method by showing that we don't need to explicitly compute the entire trie, but instead we can store and update a positional Burrows-Wheeler transform, which, as we saw in the keynote this morning, is just a few length k arrays, and then find the internal branching nodes from there. So let's take a closer look at these tries. At the second column, we've built our trie by adding the reverses of the rows, such as 0, 1 for the first sequence and 1, 1 for the second sequence. Since we've got all 4 possible length 2 sequences in our dataset, we've got a full binary tree. At the third column, we have no more repeated sequences, and so we have a unique leaf for every sequence in the trie, so we have 6 leaves. Again, we can show that we added the path 1, 0, 1 for the first sequence and the path 0, 1, 1 for the second sequence. Now, let's take a look at how we can use this trie to check for left maximality. If we keep thinking about following these sequences backwards, a branching node means that there were at least 2 sequences that were following the same path, but then diverged. Consider sequences 2 and 4. Starting from the third column, they both have value 0. But when we move to the second column, sequence 2 has value 1 and sequence 4 has value 0, which results in a branching node in the trie, indicating that the block consisting of sequences 2 and 4, starting at column 3, is indeed left maximal. Of course, we then need to check that this block is right maximal, and it would actually fail because in column 4, we can see that both sequence 2 and sequence 4 have a 0, so we could extend to the right. Now that we've seen the non-wildcard case, let's turn our attention to what happens when we've got unknown values. We might hope that we could directly extend the trie approach by adding all possible consensus patterns for each reverse sequence. Let's take a look at a simple example. Here, the reverse of sequence 1 is star 0, so we'd add both 0, 0 and 1, 0 to the try. And the reverse of sequence 2 is star 1, so we'd add 0, 1 and 1, 1. However, our definition of wildcard block requires that each sequence, excuse me, our definition of wildcard block requires that each column contain a sampled value. So sequences 1 and 2 at column 2 do not form a block. But the try has 2 internal branching nodes, which would indicate to us that there are 2 left maximal blocks at column 2. In order to account for this, we define a new try data structure, which we call the extended block try at column L. We don't explicitly construct this try, but instead implicitly explore it using a depth-first search, recording the blocks that it produces as we go. The key here is that rather than adding all consensus paths, we only add consensus paths that can be blocks, plus a few more that we end up not reporting as output. To do this, we need to keep track of which rows match to each node and do some checks as we search the try. To demonstrate how this works, let's take a look at an example. Here with wildcards and with 6 rows and 6 columns. Suppose we're currently processing the 4th column. Oh, in order to avoid adding false positive nodes, we need to make sure that we keep track of which rows match to each node. So we label nodes with the rows that match them, and we also label nodes with the path that each node represents. Remembering that this try is built over reverses, the path 0, 1 would represent a 0 in the 4th column and a 1 in the 3rd column. And to keep this picture from being too overwhelming, we haven't included the node labels for the bottom 2 levels of the tree, but they would also be there. So let's take a closer look column by column, noting that our algorithm, since it uses depth-first search, would actually not process in this sort of breadth-first way that I'll show. So our first root node has an empty path, and so trivially all rows match to it. Then when we think about adding our first column, which would be column 4, we add both of the children of the root, and we check which rows match to each of these. So for the 0 child, we would see that rows 1, 2, 3, 4, and 6 match. And for the child with the 1 path, only rows 1 and 5 match. Looking at the next column, column 3, again, we add both children of the path 0 node, and we see that path 0, 0 matches to rows 2, 3, and 4, and path 0, 1 matches to rows 1, 3, 4, and 6. And at this point, you're probably noticing that we do have a 1, 1 path in the data set, and it would be supported by both rows 1 and 5, but I haven't included it in the try here. And this is because we actually check for right maximality of the parents before adding the children. And so if we take a look in our data set, you can see that the block consisting of a 1 at column 4 is not right maximal. It could be extended right. And so we do not include its children in the try. Then finally, we would move on to the second column, adding the children of our bottommost nodes. And then finally, we'd add the last column. As I said before, instead of containing all consensus patterns in the data set, which we saw would generate false positive blocks, this try contains true blocks only, plus some additional sequences, such as that path 1 sequence that is not right maximal. and we would then not report. In the end, it turns out that all internal nodes and some leaf nodes in this construction are true blocks. And one thing to observe is that as we move from a parent to a child, the child's row label is always a proper subset of the parents. For example, the node 0, 0 has rows 2, 3, and 4, and that's a proper subset of its parent path 0. So we must lose at least one row each time we move down a level in the trie, and also by definition, we've said that each block must be supported by at least 2 rows, it must contain at least 2 sequences, and so we can observe that the maximal depth of this trie is k minus 2, and that helps derive the bound that we saw earlier. And so, as I've said, we implicitly search this trie using depth first search, only making our recursive calls to explore child nodes if they could lead to a block. And we can show that it will only take big O of kn time between blocks as we're processing the data. Thus, our overall runtime of the algorithm is big O of kn times t, the number of blocks. We tested this algorithm using a human chromosome 22 dataset with SNP calls for 5,008 individuals. This data comes from the 1000 Genomes project, and it was also used in the Alenko et al. And Cunha et al. Work that I mentioned before. Our dataset did not include any missing values, and obviously that is a really important part of this project, so we randomly assigned some SNP calls to be wildcards. As a baseline, we ran one experiment with no wildcards, denoted by 0%. In another, we set 5% of all data values to be wildcards. And in our final experiment, we set 10% to be wildcards. We also varied a minimum area parameter, where the area of a block is defined to be the number of sequences in the block times the number of SNPs in the block. And in the data I'm showing here, the rightmost 3 columns are all averages over the number of blocks found. So while we see that the total number of blocks increases dramatically as the number of wildcards that are present in the data set increases, going from 100 million to 500 million to over 1 billion in the 10% case, the total number of blocks that are over 500,000 or 2 million area grows more slowly. And in fact, if we take a look at a scatter plot showing the number of sequences per block versus the number of SNPs per block, we see that, and this is specifically for larger blocks, so at least area of 500,000, we see that the general distribution of these blocks pretty much stays the same, even as we go from no wildcards at all to 5% wildcards to 10% wildcards. So some takeaways here. First, we saw how maximal perfect haplotype blocks, both with and without wildcards, can be used to identify genetic variants in a population in order to compute selection pressure along a genome. Then we saw the first algorithm to find all wildcard blocks in particular, which runs in big O of kn times t time. We also saw experimentally that for larger blocks in the distribution, the distribution of blocks remains mostly the same even as we increase the number of wildcard SNPs. And we think that this suggests that having unknown SNP values may be workable even when we're interested in computing selection coefficients. We also suggest some open questions. Can we find better bounds on T, the number of wildcard blocks? What about a faster algorithm to compute all blocks? Finally, I note that this is ongoing work. We have given a pangenomic formulation of this problem, and we're currently exploring an application to coronavirus data. And with that, I will thank you for listening, and you can find the implementation information on this page right here. So thanks, everyone.